we truly are working on ways to change the world, to make the world a better place, cure rare and neglected diseases that startup companies and biopharma won't cure. We're working on cancer. We're working on Alzheimer's and Parkinson's. We're working on ALS, another neurodegenerative disease, Huntington's disease. These are difficult problems that millions of people around the world suffer from. And so I'm excited to say that CRISPR has a path to potentially help with all of those diseases. The livestock around the world can create up to 15% of all of the greenhouse gas emissions derived from humans. So we're working on ways to take CRISPR to actually intervene and to try to turn off and shut off that methane before it gets burnt into the atmosphere. Let's shut down the methane from cows, let's reduce the methane emissions from rice, and let's reduce the use of fertilizer so that we can really start reducing the emissions from the way that we grow and produce food. One of Jennifer's most recent breakthroughs is actually using CRISPR directly on the microbiome. So you can actually take a complex community of microorganisms and take CRISPR and make changes, precise changes in those organisms. So I think in the future you could think about potentially having a microbiome therapeutic that helps you program the microbiome to be a healthy microbiome. And who knows, maybe you could prevent these diseases from ever starting if you can go right to the microbiome and create a healthy microbiome that helps you not go down these neurodegenerative paths.
Thank you for joining Once Changing the World, India's first Future Tech meets Sustainability podcast. I'm your host, Eddie Allen, and today I'm delighted on Dr. Brad Ringeisen, a physical chemist with a PhD from the University of Wisconsin-Madison. He's a pioneer in the field of live cell printing and an experienced administrator of scientific research and product development. He was the director of the Biological Technologies Office at DARPA, where he managed a division working at the cutting edge of biology, physical sciences, and engineering. Programs in Brad's office include research in genomic editing, epigenetics, neurotechnology, food security, and biomanufacturing, as well as diagnostics and therapeutics development. Prior to DARPA, Brad ran his own research group at the U.S. Naval Research Laboratory as the head of the bioenergy and biofabrication section. Currently, Dr. Brad is the executive director of the Innovative Genomics Institute, founded by Jennifer Doudna on the University of California, Berkeley campus. The mission of IGI is to bridge revolutionary gene editing tool development to affordable and accessible solutions in human health and climate. Thank you for joining Once Changing the World. Really appreciate you taking time and being part of the podcast. Now, you spend decades at the forefront of biotechnology, from 3D printing to synthetic biology and genome editing at DARPA. For our audience, can you start by maybe explaining biotechnology, synthetic biology, and CRISPR Cas9? And how do you think these powerful tools is going to upend our healthcare industry?
Yeah, that's a great question. It's exciting. People oftentimes say that we're living in the century of biology and biotechnology, and I really do believe that that's a true statement. It started early in the century with the Human Genome Project, where there were several groups racing to try to sequence the human genome. And you can think of that project, it improved the speed and the cost of being able to sequence. And sequencing is sort of the roadmap. You can't understand a system like any system, anywhere from a microbe or a bacteria all the way up to a human. You can't understand those systems biologically until you understand the roadmap. And that's what sequencing does for you. So when those groups finally did achieve that, it was a huge milestone. And now all of a sudden, scientists had this wonderful roadmap, but it wasn't a roadmap that was labeled very well. It was very much a roadmap that had a lot of question marks and maybe some gaps and a lot of undefined areas. And so there's a lot of refinement, there's a lot of understanding, but it really was something that unlocked the rest of your question, which is synthetic biology and genome editing and CRISPR, and all of these aspects were unlocked by those early days of sequencing. So that was step one. You have to be able to understand the system before you can actually go in and use these other tools. All right. So let's go to synthetic biology. So synthetic biology really is sort of a made-up word by biologists. I think before synthetic biology, people used to say systems biology. Biology, for those of you that are not biologists, it's a really complex topic. The human system is a tremendously complex system. You have different organs that are affecting each other. You have blood that is sort of carrying different molecules and different signals throughout your entire body. And then if you go all the way down inside of a single tissue, say a lung tissue, you have different cell types. You have barrier tissues. You have the air interface. All of those things are affecting how that system changes. It responds to the environment. When you take a drug, those systems change and are modified. If you get sick, that system changes, it gets modified. And it's an entirely complex web. You pull one string of that web, and many, many, many other strings of that web are changed and modified at the same time. So I think that describes systems biology. It's that system, that web, that interconnected web, that really is the system that we're talking about. Now, when people refer to synthetic biology, they're now wanting to go into that system and make changes, cut one string of that web, add in a new string of web, that then changes the function of that system. Now, people did this obviously in more simple systems first, things like bacteria, where you could go in, it was a much smaller genome, much easier to sequence. You could understand that system much more completely, something like E. coli. And people used that, and they referred to E. coli as sort of a chassis. Just like a car is a chassis, this was a chassis that could do something for you. So people would start to go in and start making changes to that chassis to be able to see what the outputs would be. And they started doing this in a really controlled, very predictable way. You used a tremendous amount of computer power. So an entirely new field called bioinformatics arose because you needed to understand that complex web, but then now also understand what happens when you started making changes into that web. And the goal was to do lots of things, to create bioenergy, fuels, biomanufactured goods, so that normally you would use, fossil fuels or chemicals. Well, now you could potentially use E. coli to be able to make some of those materials and some of those things. And so that was sort of the beginning of the synthetic biology sort of revolution. The next ultimate step, obviously, was to go to humans, Eddie. And so that's the last step of this is, how does this now make a leap from a relatively simple system like E. coli all the way to something like a human? And that's what we're in the process of doing right now in the 2020s, because my founder of the institute, the Innovative Genomics Institute that I am involved with, was Jennifer Doudna. And she, along with Emmanuel Chapartenay, created CRISPR-Cas, the genome editing system that can now do this in humans, so that we can now start to make cuts in the human genome, add in genes to be able to cure diseases potentially, to be able to better understand human biology. And this has huge implications, obviously, to healthcare. And we can talk about that more, but that's sort of it went from sequencing all the way up now to be able to read and write into the human genome.
Doctor, really, really appreciate you kind of explaining the lay of the land of biotechnology. Yes, with the Human Genome Project, we kind of mapped out the entire human genome sequence, 3.4 billion base pairs of DNA, and now we've got the tool called CRISPR Cas9. We can actually edit out a specific DNA. So I think, yes, we are in this journey of maybe, like you mentioned, understanding the complexity of the biology or human biology. And I guess maybe once we kind of do that with the convergence of AI, able to understand bioinformatics, I think maybe we will start opening up and understanding more. And then I'm sure, that this dream of possibly maybe, biotech, maybe possibly taking the disease out of the equation. I mean, that's possibly I'm assuming it's the dream of these biotech pioneers such as yourself. So yes, I want to kind of get into that later, but I want to start with your early work at the US Naval Research Lab, on live cell printing, which was foundational to 3D bioprinting. How has this technology evolved, and how far are we from functional transplantable organs, and what do you think are still the biggest hurdles?
Yeah, that's a great question. This is a whole nother field now, where we're talking about trying to actually assemble cells. Right, our human body is created from this assembly of cells, but it's not just cells. It's complex organizations of cells. It's things called extracellular matrix (ECM) that sort of is the glue that sort of holds the cells and tissues together, and that's required because you have to have this whole complex network of vasculature that goes from, millimeter scale, well, arteries are at centimeter scales, down to vessels that are at millimeter scales, down to micro vasculature, which is down to sub-millimeter levels, down to, say, 100 micron size of vasculature. So trying to recreate those types of tissues for transplantation to try to end organ transplants, so you can actually create laboratory tissues or laboratory organs that could be transplanted into the body, is absolutely one of those holy grail dreams of biotechnologists. Unfortunately, even though I did some of my experiments over 20 years ago, we were some of the first people in the world to take living cells and to be able to reproducibly place them and print them down into patterns, vasculature networks, layers of cells, trying to create structures that could potentially heal, things like spinal cord injuries, where you have a scar tissue that you would have to sort of engineer over to be able to recreate those networks to be able to maybe heal those types of serious conditions, all the way up to things like potentially liver or kidney, or even hearts and these kinds of things. So where are we? Well, there are many technologies now that can do this, that can create three-dimensional structures of cells, pattern structures of cells, even heterogeneous structures of cells. You can create intricate scaffolds that can then be flooded with cells, and they can make these structures. But are they succeeding to trying to implant them into the body? Unfortunately, not so much right now. There's a lot of different ways to be able to do this. People are using animals like pigs to be able to sort of incubate tissues, to be able to make them more realistic, to make them mimic the human body more closely. There have actually been humans that have transplanted and received these organs that survived the surgery, but then subsequently, after a month or 2 months, they subsequently don't survive. So why is that? Well, it's I think because we still can't quite replicate the beauty of the human body. There's such intricacies. There's heterogeneity of different cell types, of different structures, of different molecules that hold those structures together. And that's one reason. And then I think the second is just rejection. How do you build? Ideally, you would want to build these tissues from your own cells so that your body would recognize those cells and not reject them. Well, that's difficult. It's difficult to scale that many cells up. It's difficult to make something on the size scale to be able to do that. Even with printing technologies, it's difficult to do that. So people are using something called induced pluripotent stem cells. These are stem cells that can be derived from adult humans, and then modified using synthetic biology, using some of the tools that we talked about earlier, like CRISPR, to be able to make them sort of shielded and protected from the human immune response. So that if you transplant them, your body sort of, it's cloaked, it's disguised, so that your body won't immediately reject that tissue, even though it's not necessarily from your body. Okay, so there's a lot of complexities. It's the structure, it's the cell types, it's creating those structures, but then it's also how your body incorporates that tissue and or rejects that tissue that are really some of the biggest problems that are facing this community right now. So I think we're probably still a decade or more, unfortunately, away from sort of seeing some of this. There have been huge advances. Some of these pig derived, they put a human organ and incubate it in a pig. I think those are probably the ones that are furthest along right now, but it still has not ultimately solved the problem.
Right, right. I hope that, we bring that timeline a little bit closer, because there's millions of patients worldwide who are waiting for implants. And if this tech is accelerated, it could just be a boon to these patients who are waiting. You have got an experience at the DARPA, specifically at the Defense Threat Reduction Agency. How concerned are you about the democratization of these biotech, tools like CRISPR and bioprinting being exploited by bad actors, to, possibly maybe, create these pathogens or bio weapons? And what safeguards do you think are most critical in the space?
Yeah, I think that it's a great question. It's probably the most popular question that I was asked at DARPA, because everyone is worried, right? With powerful technologies like CRISPR, especially, that allows you to make changes directly in the human genome, you have huge opportunity, but then of course there's huge risk as well. And I can confidently tell you right now that in order to do some of these complicated experiments, it takes entire teams of researchers to be able to do this. We need chemists that develop nanoparticles to be able to carry the CRISPR, to be able to protect it from the human immune system, to be able to target a specific tissue or a specific cell type that you want to be able to target. It takes the CRISPR engineers to be able to create the exact version of CRISPR that was able to work for the application and the purpose that you want to be able to do. And then it requires animal studies to be able to show what dose and things are needed. Someone just in their garage or somebody on their own does not have a very good probability, a very low probability of being able to create something that could incorporate all of these very complicated scientific objectives right away. So I think there's still a threat from things like state actors that can assemble scientific teams to be able to do these things. But there's always been that threat. There's always been that threat. And I think that's something that hasn't changed. Maybe the threat is more pronounced now, but it's not, it's just new tools that could still help with the same threat that's always been here. So I think that's one aspect of this. The second is sort of to think about the future. Where are we going with this technology? And I do think that we need to be careful and safeguard the technology. One of those safeguards is inherent in every single university in the world, every single lab that does this work, whether it's in a company or whether it's at a university. They have to follow protocols. It's called an internal review board, an IRB, and every person and scientist that has to work with these molecules and these tools has to get approval from their IRB. So number one, follow the rules. Everyone should follow the rules. And we have to have oversight committees to make sure that people are following the rules. And some of the notable examples of people either lying to or avoiding those IRBs are when bad things happen, like the CRISPR babies and things that had happened previously in the world, because those scientists and doctors sort of avoided the rules or weren't fully honest about the risks and the benefits of the technology. And so, first of all, honesty, transparency, and following the rules are some of the biggest safeguards that you could possibly have. Now, let's just switch to talk about the technological aspects of what you could create. I think there are some unfinished business that needs to be done in terms of building safeguards into this technology. We need better ways of sort of forensically understanding when this technology has been used. If you look at a bacteria or if you look at a human cell, how can you tell if CRISPR or other editing technologies has been used? That's something that it's sort of still an unanswered question. It's still difficult to do that. So I think building in ways to be able to sort of put a tag or a mark to be able to show that you have done something to this system, that others in the future could go back and to be able to tell that this has been a human modified system, I think would be advantageous. So I think that's one safeguard that could be done. A second safeguard would be ways to reverse an edit or a change. Can you go in the same way that you would make a modification? Can you then reverse and try to then sort of go back to the native system, to be able to go back to the previously edited system? These are technologies that also don't exist right now. Okay, so I think those are some of the safeguards that scientists still need to work on developing those types of tools.
Right. Yeah, Doctor. So I think the world is getting more and more democratized. I mean, these tools are being more freely being accessed and accessible, and plus there are these large language models. I mean, these open AIs and these Claudes and Groks, and I think just last week, Google kind of released their own AI scientist, model, an agentic AI model. So I think these democratization of tech stack, yes, it's beautiful for the world, because I mean, for the ones who could never get access, has access and can tinker around with these tech. But yes, being responsible is another thing, because there will be bad actors, because there's always been bad actors and there always will be bad actors. And I hope that, maybe like you mentioned about, maybe if we have systems which understands that, maybe this was a human modified system, I think that will be really, really cool. And I hope that, scientists, researchers such as yourself are always ahead than the bad actors.
We hope, but it's difficult sometimes to tell. And I think any organization or any person that really wants to try to do something bad will find a way to do it. But honestly, there's much easier ways to do bad things in the world than to try to CRISPR engineer someone in a nefarious or bad way. And so I think what you see is that's what's happened in the world over and over again, is that you end up having traditional sort of warfare, explosions, or other ways to do harm to people. And you don't see CRISPR being used in this nefarious way, at least at a large scale. And I think because it's really hard, it takes a lot of effort, and I think there's also an awareness of how dangerous it can be as well. And that's something that it gives people pause as well. I will just also say that our institute, the IGI, is also doing a lot of work on sort of training, both for human health and for agriculture, because this is a technology that can work on plant cells as well. And so there's huge opportunity in agriculture. In fact, we just finished a training session at ICAR in India, the Agricultural Research Institute in India. So to be able to train the next generation of scientists in using CRISPR for agriculture. We've also done several of these CRISPR training courses in Africa to train African scientists, because in order for there to be real innovation across health and climate and agriculture, you really need more and more and more people using this technology and trained in this technology and understand the power, and to do this in a responsible way. So that's one of the missions of our institute, is to go and really train and provide access to these technologies.
Right, Doctor, I want to get into IGI, but before that, wanted to kind of still, talk about DARPA. What are you most proud of from your time as director of the Biological Technologies Office, and especially regarding mRNA vaccines and COVID-19 responses?
Yeah, I mean, I was at DARPA when COVID hit, and it was just all hands on deck. Everybody wanted to help. And luckily, there were some technologies that we had invested in for sometimes upwards to 5, 6, 7 years before COVID, that really ended up making an impact in this. And one of them obviously was mRNA vaccines. We had funded Moderna back, I think, in 2012 or 2013. There was a program manager named Dan Wattendorf, who is now at the Gates Foundation. But Dan had the foresight to look at the world and understand that there were rapidly evolving viruses, these RNA viruses, that traditional vaccines that took years to develop and manufacture were not going to be an appropriate response to something like a coronavirus or an influenza virus that could potentially rapidly mutate and change and make the current existing vaccines sort of irrelevant. So he invested in a whole portfolio of technologies. He did DNA vaccines. He did mRNA vaccines. He looked at antibodies as treatments to be able to slow or stop that could be given to warfighters or citizens to be able to protect them from disease. And it turns out that a couple of those technologies really did pan out. And obviously Moderna grew, but we had seen human clinical data as early as 2017, 2018, where we were developing a chikungunya vaccine, an mRNA chikungunya vaccine. So we were fairly confident. We were seeing good safety data. We knew before COVID that mRNA vaccines were for real and that they were going to work. And then obviously you saw the accelerated timeline with things like Operation Warp Speed to then really scale and demonstrate that this could work at scale. But early on, without those early investments, I really hesitate. I don't think Moderna probably would have been able to really stay working on that technology without those early investments from DARPA. A second example is thinking about what we call platform technologies that could pivot quickly to look at something that emerges in the world. One of those platform technologies is CRISPR, clearly. But we've talked about that sort of being able to use for human cells or on bacteria or on plants and crops. Well, another one of these platform technologies involved sort of searching and trying to identify antibodies. These are molecules that are in the body that are used for signaling and for recognition. And over the past decade, they've been developed for infectious disease. You can actually develop an antibody. Your body develops antibodies that sort of binds and blocks viruses or bacteria as they invade your body. And so your body's sort of naturally and continuously doing this. And that's part of how mRNA vaccines work as well. You're sort of programming your body to create these protective monoclonal antibodies. So we had developed a platform at a company to be able to quickly take a human sample, a blood sample, and to be able to quickly isolate antibodies from people that had been infected. So some of the very first samples were given to this company so that they could sort of very quickly assess and identify and capture some of the antibodies from some of the first people that were infected with SARS-CoV-2. And it turns out that we found very, very good antibodies that bound and neutralized the SARS-CoV-2 virus. We scaled that, and then ultimately the company Eli Lilly licensed that technology and essentially created one of the first monoclonal antibodies that was neutralizing at the early stage of the pandemic. So those were two things that I was very, very proud of and very excited that we were able to contribute to. But I mean, we started programs on capturing water from air to be able to help remote villages or remote camps of warfighters to be able to capture water to drinkable, potable water so that they didn't have to carry that water with them. We started protective, very advanced ways to be able to protect the human body from chemical and biological threats. We worked on ways to stabilize blood so that you didn't have to constantly have a cold chain, freezers and refrigerators to be able to transport and preserve blood, so that if you needed it on the battlefield or not, you could have a preserved blood that then could be given in need. So there were so many different technologies at DARPA that we started. It's sort of a scientific playground that really is just an amazing sort of a crown jewel of the United States.
Oh, cool. I mean, these innovations that you're talking about, it's so cool. And yes, DARPA has been at the forefront of, creating some of the finest innovations. Now, Innovative Genomics Institute. Uh, can you talk a little bit about it? Your role into it with IGI. What nudged you to kind of transition to IGI, and what are the top scientific and strategic priorities you're overseeing at IGI?
I had been there for about almost 4 years, and I saw this opportunity. So I knew that I would have to leave at some point. I was actually able to stay there for up to 6 years because I ran the biological technologies office, and so by being in leadership, they were I had succeeded, so I could have stayed there for up to another 2 or 3 years. Um, but I saw this opportunity at the Innovative Genomics Institute. We're on the campus of the University of California at Berkeley, but we work with the University of California, San Francisco, one of the leading medical schools in the country, and the University of California, Davis, which is one of the world's leading agriculture universities in the world. So we work across all three of those universities. And it was founded by the Nobel laureate Jennifer Doudna. And when I interviewed with Jennifer, I was sold. She is inspirational. She wants to change the world with this technology. She knows that her lab alone cannot do that. And so she founded this institute to be able to create a collaborative network of scientists. It's almost like a research, juggernaut, an innovation hub that is grounded by CRISPR and genome editing, but we do much, much more. And we do it across human health, and we do it for agriculture, and we're even working on greenhouse gas emissions and ways to be able to limit greenhouse gas emissions or capture carbon and store carbon. So this technology has truly the ability to change the world. And when I saw that and I met Jennifer, I decided, okay, the time was now for me to leave DARPA and to start working at the IGI. And I've been here now for almost 5 years. And we truly are working on ways to change the world, to make the world a better place, to cure rare and neglected diseases that startup companies and biopharma won't cure. And we're trying to bring that to hundreds and thousands of people around the world. We're working on cancer. We're working on Alzheimer's and Parkinson's. We're working on ALS, another neurodegenerative disease, Huntington's disease. These are difficult problems that millions of people around the world suffer from. And so I'm excited to say that CRISPR has a path to potentially help with all of those diseases, not just rare diseases and neglected diseases, but some of these larger diseases as well. So that's part of our institute, is trying to really innovate ways to be able to develop, to see how CRISPR could either prevent or treat those diseases. But it doesn't stop there. We are, like I said, we just got back from a big contingent, got back from India. We're partnering with these research institutes, ICAR, IARA, IRE, NIPGR. We're partnering with researchers and scientists at those agricultural institutes and organizations in India, because we know there's 1.4 billion people in India. We need to be able to more sustainably and more reliably create the food and more nutritiously provide food for that population. And we're doing it in partnership. This is not something that the University of California or the IGI, we're doing it in partnership with researchers there. We need to create drought tolerant, salt tolerant, heat tolerant, disease tolerant, flood tolerant varieties of things like rice, mustard, chickpea, legumes, pulses, millets. So we're working in country to be able to help and to be able to do that and provide guidance and collaboration to be able to do that. And then separately, we're also working on livestock, cows. I'm sure you might know that livestock create a tremendous amount of methane, and methane's a very potent greenhouse gas. And when the livestock around the world can create up to 15% of all of the greenhouse gas emissions derived from humans. And that's just from sort of the livestock around the world. So this is an enormous problem. And those livestock are generating methane because of the microorganisms the microbes that are in their gut, in their rumen. It's called the rumen, the stomach of the cow, and they're burping methane back out into the atmosphere as they digest the feed and the grass and things that they're digesting along the way. So we're working on ways to take CRISPR to actually intervene and to try to turn off and shut off that methane before it gets burped into the atmosphere. So the opportunity is huge, but the world needs these technologies. They need it for human health. We need to create these cures and treatments for more and more and more people, and to do it in an affordable and accessible way. And we need to do the same for agriculture and for climate. It can't just be in the United States. We have to be able to get out into the world and to be able to affect change in countries that need some of these technologies the most.
Kudos to you and your team, and you guys have like a rock star team at IGI. And yes, I think collaboration is the future. The more you collaborate, the world is hyper connected. But I think what's happening is like most of the nations are going inwards rather than outwards. And that's happening in China, that's happening in India, and that's happening in other countries also. We live in a hyper.
We're trying to buck that trend. We are.
Yes. Yeah.
We are going out. I talked to one of our scientists, Dave Savage, who works closely with Jennifer on developing new CRISPR technologies. I think he's traveled to Africa 5 times and now India twice, just in the past 12 or 16 months. So we are sending our scientists out into the world. We're building collaborations. We're building networks, because these things are going to happen probably in rich countries like the United States. We really need them to work in lower resource environments where a lot of people are fighting climate change. They're on the front lines of climate change. They're experiencing floods. They're experiencing 50 plus degrees Celsius temperatures. These are threats to the way humans are living. And I think that this technology can help provide some solutions.
Right, Doctor, I completely double you. So I've been living in Bombay, all my life. For the last 50 years, I've been living in a city called Bombay. And I think all cities have this thing, where there's inflow of migrants coming in. Eventually, it's small piece of land, it gets overpopulated. There's constant consumption without conservation, which leads to resource depletion. And I think we're staring at a climate crisis, and it's 25th March today. For the past 3 years, I've kind of moved out from Bombay to Goa. Goa is like a tropical kind of place. And today is 25th March, and it's supposed to be peak summer season, and it's pouring cats and dogs. So yes, I think there's these disruptive weathers which is there around the world, and I think these tools such as CRISPR Cas9 is going to play a huge role. You spoke about agriculture. I want to kind of get the thoughts on the healthcare space. I mean, CRISPR has been touted as the next big thing. Specifically in the healthcare space. There are some treatment for sickle cell and so on and so forth, but these are really seriously expensive, like millions of dollars. How do you think this is going to change? When do you think this price point is going to go down, and this is going to be more accessible and democratized? Will be great if you could shine some light on that.
Well, it's a great point. I'm glad that you brought it up. It's disheartening to see a technology have the potential to transform healthcare, and you see it with the approvals of the first CRISPR cure. Essentially, I mean, it seems to alleviate and all of the symptoms of sickle cell disease, but the price point's at $2 million, and so I think there's just a handful of people that are being treated by this treatment right now. And look, you're talking about millions of people around the world that could be helped by this technology. But at $2 million a treatment, it's not possible. And honestly, if you look at how the treatment is done, it's a pretty painful and long process as well. You have to have a bone marrow biopsy or a bone marrow. You have to take the bone marrow cells out of your body. You then have to take those cells. You have to sort of purify them, find just the cells that you need, treat them with CRISPR. At the meantime, you have to treat the patient with a radiative therapy to sort of kill some of the existing cells in your body, so that the transplanted CRISPR cells can then sort of take hold and to be able to go through. So it's a months long process, and that's part of the reason it's so expensive, but it's also sort of a painful and difficult process. But the end result is very, very powerful, that the treatment seems to work and work very well. But I think by no means is that an ideal solution. An ideal solution for me would be to put the CRISPR treatment directly into your body and have it find those cells directly so that you cut out most of those steps and you just go in and essentially can make the treatment. Ideally, that could be one dose, but it is very difficult to do that. It's very difficult to program CRISPR and these nanoparticles to be able to target specific cells inside the body. It's a little bit easier to do the liver. And so I think you're going to see treatments for liver disease first, because liver is sort of the trash can of the body. Everything kind of goes to the liver. So when you inject something into the body, it'll be captured by the liver. So I think you're going to see some of those treatments for liver diseases first. But the next steps will be, can you target things like the kidney? Can you target the stem cells that are in bone marrow to be able to treat some of these blood diseases like sickle cell? And if you could do that, if you could accomplish that, and I'm seeing data to suggest that we're going to start to be able to do this within the next few years, I think that will naturally help the prices come down some. Okay, so that's technologically, I think there's going to be ways to be able to see these prices come down, but it's still going to take time. And so what you're seeing is sort of a bubble. It's sort of a CRISPR bubble right now. There's so much promise that you saw so many companies be founded in the biotech industry, but they've all done the same math. They've done the same calculus. They've said, okay, what tissue can we access? What diseases are easy to treat, meaning that you can just change one gene, and they all end up going after very, very similar diseases. So there's an economics aspect of this as well. And when you start seeing just a handful of patients being treated, $2 million price points, investors are starting to think, ooh, I don't know if there's a model here. I don't know if there's a profitable model for this technology right now. So you're starting to see a wave of some layoffs, of people that are sort of pulling back a little bit in the industry. And I think that's a natural. You expand and then you contract a little bit. And I think for me, it's saying there still needs to be technology development, which is what the IGI does. That's what we do. We develop new tools, new technologies, things like these treatments that potentially could go and just be added into the body and hunt down the exact right type of cell and achieve a high degree, a high efficiency of editing in exactly the right cell type. Those are the types of technologies that I think will break this cycle, lower costs, and to be able to provide these types of treatments for more people. But in terms of the safety and in terms of the efficacy, you're seeing very good results. And so I do think ultimately CRISPR and genome editing is going to be the future of healthcare, but it's just going to take some waves and a little bit more time and some new technology development before you really start to see it exponentially taking off.
True. Doctor, your portfolio was neurotechnology at DARPA and neurological disorders. I think it's the biggest problem, because I think you mentioned about, the biology or human body being complex, but I think the most complex part is the brain, 100 billion neurons, 100 billion synapses, and that's the black box. If you kind of are able to kind of probe it, through neurotech, CRISPR, I think, the world would completely upend. What are the neurotechnology breakthroughs in CRISPR you think are required, which might possibly open, the doors for, possibly these treatments for neurological disorders like Alzheimer's, Parkinson's, Down syndrome, and so on and so forth?
Yeah, well, I think there's some nearer term solutions and there's sort of longer term solutions. I think in the near term, out of some of our labs at the IGI, we're already seeing some large animal studies where we can actually do injections into the brain and see good sort of diffusion of CRISPR throughout the brain. And so I think that and good at editing in the brain. So I think in the near term, there are going to be ways through sort of direct injection to be able to treat specific regions of the brain and to be able to make edits. The problem is that you still need to know what to edit. And that's that complexity of the brain. There are some diseases that are, the route might be more direct. We're working on some treatments for ALS and for Huntington's. These are diseases that have some genetic backbone, some genetic causes. It's complicated still, but I do believe that there might be ways to be able to directly edit cells in the brain to be able to treat those diseases. Okay, so we're currently working towards both treatments for both of those diseases. But then if you go to the even more complex diseases, things like Alzheimer's and Parkinson's, the origins of these diseases are still somewhat unknown. We don't fully understand how a healthy person ultimately gets these diseases. There could be several complications. It could be environmental factors. It could be things that are going on in your gut, the gut microbiome. There seems to be connected to the onset of these diseases. If you can believe it, like changes in the microorganisms living in your gut, they seem to communicate with the brain. As you're digesting food, they send signals to what's called the enteric nervous system, and that enteric nervous system then communicates with your brain. And the earliest onsets, some of those signaling molecules may actually be inducing some of the onsets of these complicated diseases. So for me, there's going to be stages to go after those diseases. There are some genetic underpinnings. There are certain genes that make you more susceptible to Alzheimer's, that make you more susceptible to Parkinson's. So there are groups that are working on maybe direct edits to be able to do that. But how do you edit the entire brain to be able to affect change? That's a very difficult concept to think about doing. There are others, by the way, that are looking at making programmed stem cells and programming those stem cells to inject into the brain that then treat these diseases. So there's a lot of different ways to potentially go after that. I'll mention one very, very novel, interesting thing that's going on at the IGI. One of Jennifer's most recent breakthroughs is actually using CRISPR directly on the microbiome. So you can actually take a complex community of microorganisms and take CRISPR and make precise changes in those organisms. So I think in the future, you could think about potentially having a microbiome therapeutic that helps you program the microbiome to be a healthy microbiome. And who knows, maybe you could prevent these diseases from ever starting if you can go right to the microbiome and create a healthy microbiome that helps you not go down these neurodegenerative paths. So I wanted to at least throw that out there as an exciting new pathway that maybe in 10 years or 15 years, maybe we have these treatments that could help promote a healthy microbiome.
How awesome would that be, Doctor? Dr. Artificial intelligence, through the course of the conversation, we spoke about AI and how AI is being used. And, AI right now is possibly transitioning from, these LLMs, foundational statistic models, to, what they call agentic AI, and spoke about, how Google is possibly, worked and built this AI scientist, which they've given these access to.
And I didn't answer this question well. About AI, so I can talk specifically about AI.
Yes, please. That'll be really good. How AI is converging with biotech.
Great. So this is an important topic right now because there's a lot of excitement about. I'll put it in a positive light. There's excitement about AI and healthcare right now. Now, do I think that AI is going to be able to just create cures, whether it's a new drug or new CRISPR therapies? On its own, no. There has to be robust and large amounts of data generated, trusted data that goes to the root cause of these diseases. And you need to generate that data to be able to feed into and train artificial intelligence. So that's number one, is that one of the reasons that this new technology that just won the Nobel Prize called AlphaFold basically, if you give a sequence to this algorithm and learning model, it will then give you the structure of the protein. And the structure of a protein oftentimes is sort of the secret to the disease. A lot of times people will take a drug and specifically target a protein to try to drug that protein to try to treat the disease. But the secret to AlphaFold was that there was already a fantastic data set available to AI engineers to be able to feed and train their algorithm with this data set. That doesn't exist for diseases and other drug targets, and for engineering new CRISPR variants, or creating new drugs. The data is very sparse. There just is not enough data to really be able to reliably do this. So there's a lot of promise in AI, but you also have to match the advancements in AI with advancements in high throughput data generation for human health. So that's really step one. Step two is to now try to untangle that web that I spoke of earlier, the complexities of human disease, the complexities of the human body. I do think AI, trained on the correct data, has the ability to be able to elucidate some of those complexities, to be able to sort through the important targets and the wrong targets. So I think one of the biggest benefits, I think, for AI is that it will reduce the number of experiments that you have to do, reduce the number of animal studies that you will have to do, and to really target scientists on the right solutions, the right targets, so that now you have a smaller set of drugs to be able to then test in more detail. So it should speed the process. It should reduce the failure rates and increase the success rates. And ultimately that will save money. It might lower costs of healthcare by doing that. If you reduce the number of experiments and you speed the amount of time between discovery to a human clinical trial. That's sort of the promise. I think that's the realistic promise of AI. The concept of just inserting in some data and it just spitting out the magic drug, I think we are a long, long, long way away from being able to do that. And I think that those that are sort of promising that, I'm not sure if they understand the complexities of the human body and complexities of human disease.
Right. Thank you for highlighting that. Doctor, you spoke about, creating these cows, which could, possibly reduce these methane gas emission. You spoke about CRISPR for agriculture. Would you be able to talk about how do you see CRISPR addressing the challenge of feeding this 10 billion people? Eventually we are 8 billion and we growing. How do you see these, CRISPR food and maybe CRISPR playing a role in net zero farming and CRISPR for climate? It will be great if you can elaborate on that.
Yeah, in the near term, we're going to be talking about what I call climate resilient or climate adaptable crops. That's going to be in the near term. People are already demonstrating drought tolerance. So use growing crops with less water. You're already seeing disease tolerant crops. But these are now sort of in field trials. They have not yet really made it to a commercial product. So I think, but within the next 5 years, you're going to start to see this. The government regulations in countries around the world are becoming more favorable to genome edited crops. And so I think you're going to start to see more and more and more crops like this emerge. And what CRISPR does and what genome editing does, again, it speeds the cycle of how to create the next generation of crops. And I think that's the biggest impact, is that you're going to see discoveries get to the field and ultimately get to the market faster because of CRISPR. And remember, CRISPR is a very precise tool. It goes in and just makes, in some cases, just a single change. Oftentimes, the way foods are produced now and seeds are produced now, you make random changes throughout the genome. CRISPR doesn't do that. CRISPR is a highly precise targeted technology that I think is much safer than the way that foods over the last several decades have been created. So I want to make sure people understand how precise and safe this technology is. Okay, number one. So, and I think the earliest products that are going to hit the market are going to be things that can help foods adapt to the changing climate that we have in the world right now. And I think that's going to be important. But ultimately, I really think the biggest impact is going to be on reducing agricultural emissions. There's a Food and Agriculture Organization report that was released a few years ago that says that agricultural systems in the world are up to 30% of all human-derived emissions come from the way that we grow and produce food. What, 30%? That means transportation, energy. We could solve all of those problems, and we could still almost have 1/3 of the global emissions still exist if we don't solve the food problem. Okay? So, what are the biggest offenders there? Well, I mentioned livestock. That's upwards to 10 to 15% of emissions. Rice is also a big emitter. And I know in a country like India, it's very dependent upon rice. There's methane that's emitted from rice paddies as well. So that's a really big problem. And we're studying that problem right now. And we found a variant of rice that seems to significantly reduce methane emissions. And that's ways of sort of connecting the rice plant to the organisms that live in the soil that you grow the rice in, because the methane is being generated by organisms in the soil. So I think there are ways to engineer the crop to sort of talk to the microbes in the soil to be able to maybe reduce the amount of emissions that are being produced while you're growing rice. And then the last one is fertilizer. There was the Green Revolution, and look, I talked with the Indian scientists just a few weeks ago when I was visiting New Delhi, and they understand fertilizer, and they understand that they were part of the Green Revolution. That's one of the reasons that India can feed so many people in its country, is because of the Green Revolution. But one of the big problems with the Green Revolution is fertilizer. When you make fertilizer and chemically synthesize fertilizer, that results in emissions. When you add fertilizer to fields, it releases a compound called nitrous oxide, which is a tremendously potent greenhouse gas. So we really need to find ways to improve how crops grow with less fertilizer. And we can improve the way that they use nitrogen, which is what fertilizer creates and provides for the crops. And so I think there are ways to use CRISPR to be able to not only reduce methane from things like rice cultivation, but I think you can also use CRISPR to improve the efficiency of how crops use nitrogen, which could reduce fertilizer use as well. So that's it. Let's shut down the methane from cows. Let's reduce the methane emissions from rice, and let's reduce the use of fertilizer so that we can really start reducing the emissions from the way that we grow and produce food.
Right. What a fantastic world that would be, because I think we are staring at an existential crisis of sorts because of climate crisis, and I think we need to take these urgent steps, globally, and thanks to, people such as yourself who are kind of, finding answers of a better way of doing agriculture or production, livestock and stuff like that, So kudos to your team. Doctor, my last question to you. Will you be able to, leave our audience behind with what do you think the world is going to look like in the next 10 years, and how, what the role of CRISPR is going to be in that world, right from healthcare, in your healthcare, also neurotechnology, as well as agriculture.
I always try to talk about a message of hope. It's so difficult. I have three children that are in their 20s right now. And I know how difficult it's been. COVID, isolation, rising healthcare costs, climate crisis. The next generation is under attack constantly. with all of these things. How am I going to pay for healthcare? How are we going to exist in a more sustainable way? What is the world going to be like in 20 or 30 years if we don't start solving the climate crisis? The pressure that is mounting on the next generation is enormous. So what I try to talk about in these types of interviews, in podcasts, in presentations that I give to different communities, I try to preach a message of hope. Because a technology like CRISPR has the potential to accelerate solutions. But we need it in the hands of more and more and more scientists. We need to improve and speed these solutions. And I think it can make an impact in all of these ways that we've been talking about. Making more affordable healthcare solutions, making more farming more sustainable, and reducing some of the emissions that are going on in the world. So for me, I view this technology as truly something that can change the world and make the world a better place to live in. And I'm going to spend the rest of my career trying to work every single day to be able to make that happen. And it's not easy. Science is never easy. It takes time. It takes collaborations. It takes failures. So many failures. But you learn from those failures and you go to the next step. And that's what the IGI is about. And we're trying to sort of partner across the world so that we can try to improve and maximize the impact of what this technology can have in the future of the world.
Thank you, Doctor, for taking time and being part of the podcast. We're living at such a fantastic point in human history. For the first time, I think the world has got access of knowledge. Yeah. Yet, I think, the structure of how we have functioned, and I mean, all our institutes or banking or governance or politics has kind of divided us, and has created these barriers. I hope that, that this kind of gets erased, because we are living in a hyper-connected world. And you rightfully pointed out, the next generation are going to be really, really upset with us on how we have, pushed them right at a corner. They also have a right to breathe. We're just taking that away because of, the kind of, continuous consumption without conservation. And I hope that these tech stack like CRISPR, Cas9 creates that hope, which I think the entire world is looking for, that, possibly in the next 10 years, maybe all of the wrongs that we are doing gets corrected. I hope that you and your team play a big role in that. So thank you for taking time and being part of the podcast. And to my listeners, if you like what you see in here, then please press the subscribe button. And until next time, so you guys. Thank you, Doctor. Really, really appreciate it.
Thank you so much.
Thank you.